everyone, and welcome to today's webinar titled Using NGS for MicroRNA and Gene Expression Analysis and Integrated Bioinformatics for Pathway Discovery and Biomarker Verification. This webinar is part of the 10th Annual Genetics Virtual Week, and I am Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by QIAGEN. For more information about QIAGEN, visit them at QIAGEN.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature in the presentation. You can find this live chat located right at the side of your screen on the right side. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. I now present today's speaker, Dr. Samuel Rulli, a Senior Global Product Manager in RNA Profiling at QIAGEN. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Rulli, welcome. You may now begin your presentation.
For your talk. Thank you, Joby, and welcome to everybody who's joining us today. It's my pleasure to come here and give a webinar today about the exciting world of microRNA, gene expression, and looking at the intersection between those. Before I get started, I'd like to take a quick poll. You can just type the answer into the questions box. How many of us are getting started with microRNA or are already looking at microRNA? So if you're using microRNA's techniques in your lab or thinking of that, just type microRNA into the questions box. If you haven't started yet, just type in, I haven't started or getting ready to. And this will allow me to kind of customize the webinar to those who are here in attendance. Excellent. So I can see like right off the start that a lot of us are looking at microRNA and some of us are thinking of getting started. So what I'm going to do today is kind of breeze through the introduction slides of what microRNA is and why researchers want to look at it. And what I want to do is really get down into why we do the kind of microRNA library prep kits that we do here at QIAGEN, some of the features of that. And then I'd like to dive a little bit into the data analysis website that we made to really make microRNA profiling a lot easier. For anyone who's working in a lab and has gone through it and done an NGS experiment, you know that there's a lot of different steps and you may not be in control of all those steps. And sometimes that could be frustrating, but moreover, that can actually lead to a long time between when you first get your samples and when you're actually looking at your differentially expressed microRNAs. And as a lot of us know that are working in the lab, that's not even the end game. The end game is actually once we've gotten our NGS data, often we need to go back and perform more experiments. We might want to knock down the microRNA. We might want to overexpress it. We might want to go through and look at the genes, look at the gene expression component, put that together. And then we have to think about verification because even though NGS is the best thing in the world, a lot of times we're going to be asked, can we go through and show with a real-time PCR assay that at least some of our data correlates between NGS and what we're seeing? So with that in mind, I'm going to quickly start our presentation here today. This is a quick legal disclaimer just saying that the products that we're talking about today are molecular biology applications and they're not intended for the diagnosis, prevention, or treatment of a disease. If you need any of that legal specific disclaimer stuff, look in the kit handbook. You can get it at QIAGEN.com. If you're using a QIAGEN distributor, go to your distributor and they can help you find that. And lastly, if you have any questions, contact QIAGEN technical support. They can answer those as well. So this is kind of the agenda I put together today. Quickly, we're going to talk a little bit about microRNA and the background behind it. We're going to look at microRNA profiling using NGS. I'm going to talk a little bit about isolation kits. a lot about library construction and some of the features that we built into our QIAGEN library kit. And then what I want to do is I'm going to attempt to show you kind of the data analysis feature that we recently completed. It allows you to take your FASTQ files that you get back from your core facility, or if you have your own sequencing machine, upload them up into our cloud system. And what it will do is it will go through, do the read alignments, and it'll allow you to map the microRNAs, the isomers, and download that. But it also gives you a dashboard where we can look through and look at differential expression, heat maps, and then from there we can quickly find our downstream assays if we want to do verification. I'm going to talk a little bit about gene expression at the end if we have time. The important thing here is that there is a functional consequence, hopefully, of the microRNAs that are being differentially expressed. And what is becoming more and more prevalent is those associations really are driving a lot of the disease or a lot of the phenotypes that we see. So changes in microRNA, whether the gene is being up or down regulated, or on the genomic level, whether or not it has been deleted, will actually have a big impact on the gene expression stuff, and we want to see those correlations. So the important thing here is when we think about isolation at the beginning, if we're doing isolation of microRNAs, we are also capturing the high molecular weight RNA that's our gene expression stuff, and so we can come back to those samples later on. MicroRNAs are a master regulator of gene expression. Where researchers really get interested in microRNA is when they're looking at some of the epigenetic ways gene expression can be regulated. So microRNAs, and when we're thinking about the mature microRNA, they're about 22 nucleotides in size. And depending on the isomer you might be looking at, they can range from about 18 to 25. The important thing around these microRNAs is that they are processed through several different steps. There is a microRNA or a primary microRNA that after processing is going to get down into the small RNA that gets loaded into the RISC complex and then is going to regulate gene expression some way. MicroRNA have been shown to play really diverse roles in all sorts of different pathways. There's definitely a role in cancer, and they're emerging as a very important biomarker, especially in what I would call cell-free biofluids. So I've seen urine, serum, plasma, anywhere where you can think about pulling a sample, there's potential to find a microRNA biomarker. Right now, we have about 2,600 mature microRNAs that are in mir-based version 22. And so we've kind of gotten through the first years of the discovery of microRNAs, and what we're really looking at is the vital role that microRNAs are playing. And as we start to identify these microRNA, mRNAs, and these functional interactions, we are starting to see the first therapeutic applications where there is potential to overexpress or remove a microRNA and have a change in a specific type of sample. For our read alignments and our NGS data, we use the mir-base as one of the databases where we take those raw RNA or the raw sequencing reads and we try to align them back to the microRNA. So we're using version 22 of the mir-base, which has 2654 mature human microRNAs. In addition, with the kits that we're talking about, we can also get other types of RNA fragments. So piRNAs, mRNA fragments, tRNA fragments. These are all things that we can analyze with the kits that we're looking at. When we're looking at the alignment criteria around the pipelines that we're using today, we actually have a couple of different pipelines. We're going to focus on our RNA-seq analysis portal, but we do have another pipeline set up that will give you not only microRNAs, but it will give you piRNAs and RNA fragments. Another important thing when we're talking about NGS data analysis is the species that we may be working on. MicroRNA are almost ubiquitous across almost all the organisms that we usually study within the laboratory, or at least all eukaryotic organisms. And usually when we think about aligning back, human, mouse, rat have been very well annotated. There are a plethora of other organisms that are pretty good. If you're working on something that is outside of some of the commonly used species or something that may be well annotated within the mir-base, we also often take those sorts of things and just align them back to the entire mir-base and then use that as a way to predict what microRNAs may be there. With microRNAs, and with there being about 2,600 human microRNAs, it's now become kind of the workflow to use NGS first as the screening, then decide on which microRNAs are going to be verified by real-time PCR. When we think about this verification step, it's really important to think about the starting material that we're working on. In the past, we've used real-time PCR or looked at the differential fold change regulation with the idea that we would need to identify housekeeping microRNAs or small RNAs as a way to do the analysis with traditional real-time PCR. However, using digital PCR, we don't need to have housekeeping genes. And what we've seen over the last couple of years is that researchers who are working on biofluids, where it's often very hard to identify a normalization gene, have taken their NGS data to digital PCR for verification and for point-of-care testing, because now we can look at the absolute number of copies or the number of molecules that are there and not worry about the normalization strategy. When we're quantitating microRNA, there's a lot of different questions that we can answer with this. We can use microRNA, especially when we're thinking about looking at cancers, to differentiate cancer tissue from normal tissue based on the signature. We can use microRNA and build a microRNA-based classifier to identify a tissue of origin for cancers of unknown primaries. We can use microRNAs to stratify or distinguish different tumor subtypes. We can use microRNAs to predict cancer predisposition. And if we start to look at profiling microRNA out of circulating blood, tumor-derived exosomal compartments, or even looking in some of the other places, we can detect early cancers by looking at the sensitive release of microRNAs. And of course, there's no really big surprise that microRNAs are playing a role in cancer, and that there isn't a plethora of papers that have come out recently looking at the role of microRNA and COVID-19. So there are a lot of microRNAs that play really key roles in regulating immune function. And there are studies now being done on how microRNAs may have changed as a function of COVID. There's microRNAs looking at in people that have long COVID versus that have recovered quickly, and other factors around COVID research. And if we just look at the number of papers that have come out over the last couple of years, here's just a quick Google Scholar look at the number of papers, and this was done early in 2020, that there already were 207 publications, either in press or coming out, that looked at microRNA, SARS-CoV-2, or COVID. Some of the first papers that came out were purely bioinformatically driven, where they looked at the potential of microRNA to regulate different transcripts from SARS-CoV-2. And then there's now come out hard evidence of microRNAs that could play a role or be a biomarker for different types of infection. There's even a couple of studies out there that have shown that out of saliva samples, you could distinguish whether somebody had a SARS infection versus a flu infection. And if we look at the publication record over the last couple of years, this is just looking in PubMed and pulling out keywords and looking at the number of publications. If you look at microRNA in plasma, microRNA in serum, microRNA in saliva, urine, or microRNA as a biomarker, you can see that we have almost an exponential increase in the number of publications that have come out, especially when you look at microRNA and saliva. The number of increased publications, this is really being driven by people looking at microRNA, saliva, and SARS-CoV-2. If we want to go through and do profiling, where do we start? The first place to start is with our samples. And I can tell you that if you're thinking of doing a microRNA biomarker study, often the most important thing is getting your samples and having control over your samples and thinking about the number of samples that you need to go forward with. The most basic experiments for microRNA profiling really don't need a lot of samples. We can start with a limited number. We can go through and profile all the microRNAs there. And then we can think about taking the most maybe differentially expressed ones with a good p-value and moving that into a real-time PCR or digital PCR test. But we'll still need more samples to go through and to verify that. When we're looking at isolation, QIAGEN has a series of different kits that are branded under Mir and Easy, We have different kits for different types of samples. So we have one specifically for FFPE. We have one for tissues and cells, and we call the ones that are advanced are phenol-free extractions. We also have ones for both serum and plasma, and then we have ways that we can collect exosomes and enrich the microRNA out of them. One of the important things here is that if we're starting a microRNA project, with these Milanese isolation kits, we're going to get all of the RNA from about 18 bases up to the highest molecular weight that you can. And this means that those RNA extractions that we have, we can look at both microRNA and gene expression. If you have previously isolated RNA sitting in your freezer, and you isolated it with a kit that didn't get down to the low microRNAs, then obviously we can't use that for microRNA analysis. So if you traditionally isolated something using like a QIAGEN RNA kit, the cutoff of those kits is about 200 bases of RNA. If you tried using a different method, just make sure that if you go back to those samples, you have the microRNA there. Another really important thing is if we're looking at serum and plasma applications, stay away from heparin collection tubes. Heparin is a strong inhibitor of reverse transcriptases. If you do have samples that were collected in heparin, we do have protocols to remove the heparin and to allow you to get a much better reverse transcription reaction, which will allow you to get much more sensitivity and detection of microRNAs from your sample. When we're looking at the potential of using circulating microRNAs, and when I showed you those publications, the idea around circulating microRNAs is that we can take a blood draw and start to profile it, whether or not there are particular microRNAs that may be have been identified as early cancer microRNAs, or if we see a change in microRNAs, this can reveal that there's something going on. There is a lot of different things in a typical blood sample. You have all of your different blood cells. These blood cells also harbor microRNAs within them, and so we have to be careful that we do not have blood cell lysis. So for instance, if you have erythrocyte lysis, you will get a series of microRNAs that are actually indicative of hemolysis. And they're not biomarkers, they're indicative of a bad sample that you started with. There are a series of microRNAs that have been really well classified as hemolytic markers. We actually use those in some of the projects that we work on, and we do have panels of those as PCR panels to test samples before we use those. And if we identify hemolytic markers, we know that these samples are going to behave probably a little bit differently than ones in which we don't identify. It doesn't mean that the samples are bad. It just means that we have to understand that when we put the data together, these samples with the hemolytic markers may have bled microRNAs that are cellular and not necessarily things that are in circulation. MicroRNAs can also be found in different compartments when we think about the acellular part. So microRNAs can be found within exosomes, complex to different proteins, or in different types of cholesterol type compartments. The important thing here is that when you start your project, you keep the same protocol throughout. One of the ideas around working with exosomes is that exosomes are able to kind of preserve the cellular components of where they're coming from. In addition, exosomes can be specifically enriched and then classified based on the protein markers of the exosomes. So there are researchers that are going out there and enriching for the exosomes and then extracting the RNA out of that, classifying based on the different protein markers, and using that as the definition of the sample that they're starting with. One of the nice things about microRNAs as a biomarker is that microRNAs can be found in a lot of biofluids, and they tend to be very resistant to degradation because either they're complex with something or too small to be found by the RNases. They're resistant to freeze-thaw cycles, and they tend to be very stable. So even though there are samples out there where you probably aren't able to find high molecular weight genes for gene expression analysis, the microRNA component of that can still be there. And there's been a lot of research that's gone on over the last couple of years of digging through different cohorts of samples where the gene expression component is basically not there, but there is a lot of microRNA data. When we think about the biofluids and the different types of sample that we're working with, the most common sample that I've seen is blood. Second most common one that I've seen people work with is urine. Third most common but growing is saliva. And then we do have people that will do cerebral spinal fluid or look for other places where they can get fluid, like from the eye and other places. If we're focusing on blood, it's important that blood is collected and stored, and it's compatible with our downstream molecular analysis. And this really means to avoid heparin, as I said earlier, because this heparin is a very strong inhibitor of the RNA applications. What heparin basically does is it inhibits the reverse transcription process, and if the reverse transcription process works at all, we can see that the number of microRNAs and the cDNA yield and the library yield is usually lower. If you're working with samples with a collaborator, verify that they haven't used any heparin-treated samples. If you run into a problem where all of a sudden a batch of samples don't work, this is usually where we start our troubleshooting. And we'll treat the samples with a heparinase and a protocol that we have, and this often removes the heparin inhibition and allows us to go back and get some good data. Plasma and serum samples that contain citrus and EDTA are really good places to start. And the other thing is really try to prevent any set of cell lysis either during collection and storage, because cell lysis will release a lot of microRNAs. We know what they are, but it really does confound the data analysis. When we're looking at types of exosomal enrichment, there's a couple of different kits from QIAGEN. We have our Miranese Serum and Plasma Advanced Kit. This works as a phenol-free protocol. It is a really quick protocol to go through. We basically make a serum or plasma fraction, extract the microRNA, and then elute it off of our columns. If we're starting with exosomes, we have the ExoRNase MIDI and Maxi kits. This is a kit that allows us to go through and do RNA isolation exclusively from exosomes. It uses a really simple protocol where we can start with blood. We go through and we separate out the serum or plasma. We do a quick filtration of this to remove large particulate matter. We load the exosomes onto a membrane. We wash the membrane, and then we can lyse the exosomes off the membrane and then go into a traditional RNA isolation. We use a phenylchloroform extraction, remove the aqueous phase, bind it to a column, wash, and then elute the microRNA. From start to finish, it's about 20 minutes from your starting sample until you have the microvesicles isolated. After that, it's only about 35 minutes to go from microvesicles until you have the RNA isolated. The microvesicles that you capture on these exosome enrichment type membranes can be used for other functional analysis, or they can also be lysed and the RNA extracted. For our biomarker discovery experiments, when we're going through and we're thinking about starting with our exosomes or our plasma and serum, we can start with a couple of different volumes based on the sample type that we have. So if we're working with humans, we often can start with about 4 milliliters of serum or plasma. If we're working with animals like mice, we can often work with about 1 milliliter or less. And a milliliter may actually be several mice pooled together. Once we've gone through and we've enriched for our RNA, our sample QC that we start to incorporate into our samples is to add a spiking control. With our spiking control, we're going to spike in a certain number of control RNAs. So our spike and pull actually contains 52 different RNA fragments that are not homologous, or are not the same as the microRNA that we're starting with. When we spike these controls in here, they allow us to go through and to look at all of the downstream processing steps. We also have controls that we can spike directly into our sample to monitor the extraction efficiency. And then we also have ways that we can monitor certain microRNAs for whether or not the sample has lysed or not. The reason why I bring up all these different controls is that you can see each of these bubbles here. When we have each of these bubbles as a major workflow step, if we have any sort of change in the efficiency, for example here, when we're collecting our blood and separating the serum or plasma, when we go down and we purify the exosomes from here, as we go through and we purify the RNA, as we go through and we do the library preparation, By the time that we get to our sequencing step, we've had several steps upstream of that, which can be variables in our data that we're generating here. Where this variability often shows up is in the number of counts that we get in our library. Because as we go through and we do this NGS library preparation, we're taking the RNA, making cDNA, going through the different library steps, which often include bead cleanup steps, and then we're getting a library at the end that we amplified, and then we're trying to pool this together at equal amounts with all the other samples and putting on the sequencing machine. Once we get it to our sequencing machine, then downstream of this, the data analysis is pretty straight. We do adapter trimming, read mapping, and then we're going to count the microRNAs. Some of the different features that we built into our kits are really to overcome a lot of the challenges with what we see with microRNA sequencing and downstream verification. So with our kits, we don't need a lot of microRNA to start with. In the old days, and I'll say this probably about the old days being a couple of years ago, we needed a lot of RNA to start with, often a microgram or more, because we were starting with kits that had a step in it where we needed to do gel isolation. So we've completely removed that from our kit, and we can usually start with down to about 500 picograms to 1 nanogram of total RNA and build a library in a gel-free workflow. The other problem that we have is that in a lot of our samples, microRNA isn't the only small RNA that is there. So if you've ever done biofluid profiling and you've looked at serum or plasma samples, you'll find that microRNA probably only makes up a very small percentage of the RNA. You have microRNAs, you have piRNAs, you have H4 and yRNAs that are in there, you have other RNA fragments. So one of the things that we've done is we've come up with some size selection steps and some tricks in our chemistry to try to enrich as much of the microRNA that we can get out of there. So that often you can see about 2 to 4 times more microRNA reads with our kit. Adapter dimers are a big problem. Adapter dimers are when you ligate them on, the adapter dimers sometimes will self-ligate or self-amplify. We actually block adapter dimers using a novel kind of method that we've come up with. And we block these adapter dimers in order to allow us to start with less RNA into our samples. That makes it much more compatible with biofluids. If you've ever looked at the much of RNA that you get out of a biofluid, we often can't quantitate that. And so with our kit, we actually start with about 7 microliters that comes off of one of our microRNA columns. And then we go through and we just use that as a way to build the library. Another problem that we often have is that when we're going through, and if you look at a lot of the kits on the market, the number of samples that you can multiplex together is really dependent on the number of indexes you have. And if you're using a patterned flow cell, sometimes you want to use dual indexing and you want to use unique dual indexing. So one of the things that we've recently released is 768 unique dual indexes. And this means that for microRNA projects, we can now run 768 samples in a single flow cell. If you're working on a biomarker project, a lot of these biomarkers really come from having a statistical argument that you have microRNAs that are different between the samples you're looking at. When we start our experiments and our pilot experiments, we might start and go through and do 3 or 6. That's a way to allow us to go through and identify something. But if you're doing a biomarker project, often we want to have numbers in the tens to hundreds to thousands of samples. We want to have as wide of a cohort as possible because the idea is we want to develop out as universal a biomarker to stratify the sample classes that we're looking at. So having the ability to use unique dual indexing, this means that if we use a patterned flow cell, we no longer have index hopping, and having a very large number of these means that we can build a lot of libraries from serum, plasma, and biofluid samples. These are readily available samples that you can get from a blood bank or collaborate with somebody doing clinical research or seeing patients, and you can quickly accumulate hundreds of samples very quickly in your studies. With our microRNA library kit, these are the steps that you take to build a library. We start with our samples. So whether it's something that comes from a tissue, something that we've isolated out of biofluid. And what we're doing is we're capturing the RNAs that have a 5' phosphate and a 3' hydroxyl group. It needs to be a 3' open hydroxyl group. So the first thing we're going to do is we're going to do a ligation step. And we're going to add on a 3' adapter here. And this 3' adapter is pre-adenylated. So there's a poly A sequence in here that we're going to bind an oligo DT to. With this ligation step, it's different than other ways of building a library where you might use a poly A kind of process where you add an artificial poly A tail. And the reason why we do this ligation is that this makes the kit universal to almost any sort of microRNA, whether it comes from eukaryotes or something from the plant kingdom or something else. Sometimes on the 2' position, that can be modified. And if the 2' position is modified, what it does is it blocks the enzyme that will add that poly A tail that we use for the priming. So by having this 3' ligation, it doesn't matter if our microRNA is modified on the 3' side. The next thing we're going to do is we're going to ligate something on the 5' side. So we need to have a phosphate here. If you're working on samples where the phosphate has been removed, we can simply add that back enzymatically. There have been some papers that have recently been published that look at circulating RNA and say that some of the circulating RNA may not have a phosphate. And this may be an important kind of clue into something that isn't working. After we've gone through and we've made this molecule, so now we have the 3' adapter and the 5' adapter, we can go through and do the reverse transcription step here. Now, our first major thing that we do here, you see where it says block H4 yRNA. If you're working with stuff that's coming from blood, serum, plasma, some other compartments of the body, H4 yRNA is like a ribosomal RNA in the world of microRNA sequencing. It's very abundant. We don't actually know if it has a function, but what it will do is it will eat up all the reads in your library. So we use a fast select type method here, and we block the H4 yRNA out of our library, and that often saves us probably about 80% of the reads that would have been wasted with a typical serum plasma kit. Next thing, when we do this reverse transcription reaction, we have an oligo DT primer, and the primer we're using has a unique molecular index. A UMI is a random sequence. We use a 12-base UMI, and that means that every time we initiate this reverse transcription reaction, which presumably is once per RNA that we're working on, this means that that incorporates this unique tag into that sequence. So this UMI plays a really critical role when we're starting with low input samples. So anything serum, plasma, stuff we can't measure. And it allows us to go back and to correct sequencing errors. It also allows us to look at how many times we resequence the same RNA. So in situations where we don't have a lot of RNA and we start with 5 million reads, we can go back after the first time that we read those sort of samples and decide, do we need to sequence deeper to find more microRNAs if that's what we're interested in, or did we cover the complete library with 5 million reads? This way allows us to kind of gauge the reads per the type of sample that we're working on. We can always go back and sequence a library again, but often we find that if we don't need to, there's no need to waste the money. After we go through and we've done this reverse transcription reaction, the cleanup steps downstream of this is using your traditional beads. There's no need to do a gel purification because we block the adapter dimers during this reaction. And using traditional bead cleanup means that you can automate this on any sort of standard instrument that you're already using in your lab. Or it also means that this becomes highly reproducible because we don't have to go into the dark room, put the UV lamp on, and cut anything out of a gel. Lastly, for indexing, we use a PCR type indexing. And at this point, we're incorporating in the indexes that will be specific for your platform. So whether it's Thermo Fisher or Illumina, most sequencing that I see comes off of Illumina platform. And this is where you can pick the indexes that you're using. If you're not familiar with unique molecular indexes, this is kind of a little pictogram to kind of show you how they're working here. So here we have our original sample, and we have 3 microRNAs versus 2 microRNAs. When we go through and we add the UMIs, you can see each different cDNA is getting a unique color. If we went through and we did quantification based on the raw reads, any sort of PCR bias or amplification bias or sequencing bias that happens, if you count the number of reads here, we've amplified this through the process, but they're no longer a 3-to-2 ratio. Now it's just a 2-to-1 ratio. But if we go back and we count the number of UMIs here, we can take all of these molecules and we can look at the UMIs that were there, and we can count the number of different UMIs. And if we go back, we see we have 3 UMIs versus 2 UMI. And this is how the UMIs are really being used to increase the fidelity of the sequencing data. Here we have an example of some RNA that was extracted from a biofluid. On the y-axis here, we have the different number of reads. And on the x-axis here, we have the RNA input. So 100 nanograms, 10 nanogram, and 1 nanogram. And the idea here is that as we dilute down the amount of input tenfold, we should see a reduction in the number of reads that we have. When we look at the total reads, you can see that the total reads are the same. When we look at the raw microRNA reads, we really don't get that tenfold step down. And this is because when we're looking at the amplification of these libraries, the amplification may be different, or the amount that gets pulled together may be different. However, as we go through and we look at the UMI component of this and we deduplicate any PCR errors that may have happened, you can see here that we get the tenfold step down in the red. The value of the UMIs is much more valuable when we're starting with low input samples. So usually we recommend to use UMIs anytime we're doing something out of a biofluid, because there usually isn't a lot of stuff. However, if you're starting with even tissue material, this may have a lot of value for you if you're looking at low expressed microRNAs, or you're going through and using an application that may use the microRNA kit, such as you can use the kit for ribosomal footprinting, for example, or looking at microRNAs in a protein complex or other RNAs in a protein complex. These UMIs can really help when you have a low amount of analyte to start with. Just as a quick why we use a 12-base UMI, there's a couple of papers that have been published about how many UMIs you need to see a certain complexity of your sample or a certain number of RNAs. We use a 12-base UMI because this allows us to go through and cover about 64 million different combinations of sequence per UMI. And so if you're using anything less than 12 UMIs, there was a nice paper that came out in Scientific Reports that say that you need at least 8 bases. With a 12-base UMI, this also allows us to go through and do some, have a Hamming distance, and also work on a little bit of the fidelity of the sequencing and make sure the UMIs don't get corrupted during the sequencing. If you're using less than 8 bases for a serum and plasma application, it's basically not enough UMIs to have the diversity for the number of molecules that you may be starting with, especially if you're starting with a nanogram or more of total RNA. As an example of blocking and removing certain types of RNAs and what this can do for your library performance here is H4Y blocked versus non-blocked libraries. On the left-hand side here, we have 2 samples that have H4Y blocked. On the right-hand side, 2 samples where H4Y is not blocked. If we look at the total number of reads here in the top, you can see we did about 2 to 3 million reads per sample. If we look at the bottom down here, we can look at the percent of microRNA mapping. And you can see when we block H4Y, we get about 40% of our reads will map to microRNA. When we look at H4Y not blocked, we have about 25% of our reads that map to microRNA. And we look at the other RNA reads that mapped, you can see now that we only have 2% mapping to other types of RNA, and we have 36 to 40% of this mapping to other RNAs. So to make a long story short, by going through and removing H4Y, this increases our ability to squeeze more RNA molecules into our library. It means that the percentage of RNA that maps to microRNA, which is usually the analyte that we're looking at, is there. It also gives us the ability to map out other RNAs that may be there as well. So you can also see that we're able to increase the number of piRNAs, some of the tRNA fragments, and other mRNA fragments that may be there as well. So this is actually a real big value for anybody that's going through and looking at serum plasma applications. For sample indexing, I commented that having the ability to use unique dual indexing really increases the confidence that we have in our data. It means that no library is going to have a combination of indexes that are repeated. So the I5s and I7s on the Illumina platform are distinct combinations for that library. And we don't have to worry about any index hopping or bleed through on the indexes. With the single index system that we used to use, we have a couple of different flavors of these. We have a 96 well plates that come as lyophilized or liquid. This is often the decision that you make whether you're doing it manual or on a robot. Recently, we released 768 indexes. They come in different combinations. They're 96 well plates. So we have 8 different 96 well plates. These come as a liquid with a pierceable foil seal. These are in a very hard plastic plate that you can pick up and use with your robots. And this is what we use when we talk about automation of the microRNA library kit. We also have different versions of the core enzyme component. We have a 12 kit, a 96 kit, and then we have a 384 sample kit that we call our automation kit. If you're thinking of automating the QIAseq microRNA library kit, this has been done on almost every robot in the market. We often start by going through and using the smaller 96 size, but when we get to high volume sequencing, we use the 384 automation kit because we've increased the amount of reagents so that we can account for the dead volume that you may have. For data analysis, we developed out a website to help with this because a lot of researchers who are getting into microRNA profiling, this isn't a full-time thing you may be doing. It's exciting because you probably have samples, but to go through and to find a bioinformatics scientist who's interested in helping you, or if you've done a project and you want to go back and remove samples and redo differential expression, this can be very tedious. So what I'm going to do here is I'm going to switch from talking about this to actually giving you a demo of what this looks like. Let's see here and pull up my demo page here. Okay, so hopefully everyone can see now what looks like a different presentation. Can everyone see the where it says Demo Project Wrap 2.0 in the upper left-hand corner and some tiles on my screen here? All right, so I can see at least a few of you out there can see our RNA-seq analysis portal. So the way that this works is when you get your FASTQ files, either they're sent to you or you get them somehow in a database or a cloud environment, you can upload them to this portal. Or if you have access to base space, we can directly pull the base space files into this environment. In this is my kind of experiment folder on the left-hand side. And so you can see I have microRNA here. But I also have some gene expression experiments here as well. This is one project I might be working on here. When I uploaded my microRNA samples, I told it to align it to mirror-based version 22, and this is a human sample here. And I have 2 tiles that I've made. The tile on the right-hand side is my samples and quality control. And so typically the first thing that we look at is we go through and we look at the samples that we have. And what we're looking at here is a PCA plot. And on the left-hand side here, these are the samples that are from a liver sample, and on the right-hand side, these are samples from a kidney. And you can see here that according to the tissue, they cluster together. So this is something I would expect. The difference on the y-axis here is actually probably has to do with the sequencing depth that we have. From here, I can actually download the microRNA values or the isomer counts. So with our mapping that we use, one of the things that I really didn't talk about too much, is more of a technical thing, is that when we ligate those adapters on, the ligated adapters have a fixed sequence. And the reason why we use a fixed sequence is because this allows us to know what is there, and we can identify isomers on both ends of the microRNAs. So these are small bases that are different on the microRNA family that we're looking at there. Isomers are becoming more and more important, and the ability to identify different isomers probably will turn out to be an important thing when we start to think about how these microRNAs are being processed, or when we start to look at maybe other types of RNAs that may be processed through the same thing, such as CRISPR process or something like that. When we look at quality control, this has all the information from the sequencing run, and so you can go through and look at this. It gives you the number of UMI reads, how much of this was annotated, and as you scroll down here, you'll get into all the different biotypes that were found, piRNAs, tRNAs, and everything that is there. After we go through and we are happy with the QC, and this is something you can do by yourself, we go back and we can look at the differential expression. And this is where a lot of biologists really should be spending their time. Now we've done an experiment, we have two different types of samples here. And what we're looking at here in the center part of the screen is our differentially expressed microRNAs. On the right-hand side over here, we have a heat map. Upper left-hand corner here, we have the features, which is actually the microRNA, and we provide both the false discovery rate p-value and a standard p-value. And then down here on the bottom, we have some really interesting information here, which is coming from Ingenuity pathway analysis. So we have canonical pathways, upstream regulators, and diseases or functions that these microRNAs may look at. For this sample, it's a very basic sample, so I don't know how much value we're going to get out of Ingenuity at this point. I'll show you what it looks like with a gene expression component. But the idea here is if we want to develop a biomarker, the most important microRNAs are the ones that can tell us which of our samples are different. So what we can do here is we can actually start filtering the data here along the x-axis. We can actually increase the p-value here, and we can get down to a much smaller set of microRNAs. And now you can see from the heat map here, our samples are clustering kind of together here. We got the tissue versus the kidney. The blue and the red is the relative expression values there. So if we wanted to differentiate between the green and the purple samples here, we have two microRNAs here that are low expressed and high expressed in the other, and we have a couple of microRNAs down here that are reversed, which, you know, if we run more samples, this may actually be a nice little panel that we could use. And if we didn't know what the sample was, if we looked for this pattern, we could actually tell whether it was from one of these different tissues. One of the nice things about this is once we've identified these microRNAs, if I click on Save and Go, it saves the data, and then it takes us to the GeneGlobe Assay hub, which allows us to identify the PCR assays that we'd want to use for downstream verification. And once you've gotten in here, we have the ability to build custom panels, and then we can also find ready-to-order products, such as the individual microRNAs or panels or anything like that. So this is a really quick way to go and to do NGS, have a data analysis tool, and then to find your downstream PCR things to go through and look at more particular samples that you were interested in verifying. When we think about gene expression, so if we think back to the beginning, if we want to correlate microRNA with gene expression, microRNA regulates genes. If we see a microRNA up, its correspondingly regulated gene might be downregulated. Vice versa, if we see a microRNA down, we can predict what genes may be regulated. We might see genes that go up. If we think about that gene expression stuff, we can basically take the same workflow. We've isolated the total RNA using the Miranese kits. We can take an aliquot of that. And then what becomes important is what type of library prep are we going to use? Are we going to look at using a stranded kit, or are we going to go through and use a 3' sequencing kit? So let me slide my slides back over here for a second and reverse this. So identifying the gene expression component allows us to go through and not only find microRNA. So if you're working in serum, plasma, biofluids, a lot of the times the gene expression component is not there. The microRNA is the end story, and your challenge often is in taking that NGS assay, is whittling it down to the bare minimum number of markers that you can put onto a digital PCR platform. However, if we're starting with cells and we want to look at the molecular mechanism of what microRNA is doing, that gene expression component can be critical for us going through and making these functional consequences. It will allow us to identify which genes are being up and down regulated, look at the pathways. We can manipulate that by knocking out the gene, upregulating the gene, changing the microRNA. For the gene expression component, there's basically two major ways that we can do this. One is to use what I call a stranded RNA-seq kit. And this is where we can go through and decide on whether we want to only sequence the mRNA or if we want to sequence mRNA and long non-coding RNA, and really where the poly A tail is going to be optional. If we're starting with an mRNA capture, we often start with 100 nanograms of total RNA. We enrich for the mRNA, which is probably 1%. which leaves us with about a nanogram of enriched RNA that we'll use to build our library, and we use about 20 million reads per sample to do the sequencing. If we're looking at the mRNA, long non-coding RNA, and where the poly A tail may not be there, we use a method where we block the ribosomal RNA. We often can start with a lot less sample, so maybe a nanogram to 20 nanograms, and we use about 50 million reads per sample. Another way that we can make the association is to use 3' sequencing. And over the last couple of years, 3' sequencing has really become a nice way to do a quick survey of the gene expression component. You're looking at the RNAs that have a poly A tail. 3' sequencing tends to have a much more sensitive way of collecting the mRNA. So we often can start down to anywhere around a few cells or a single cell up to about 10 nanograms. And the nice thing about 3' sequencing is that we're not sequencing the entire gene. We just want to know whether or not the gene is up or down regulated in our sample. And we don't need to sequencing anything more than that 3' region. So instead of using 50 million or 20 million reads, we can usually get away with about 2 million reads. So for researchers that are thinking of doing that gene expression part, you do have the RNA there. If you've isolated it, you just have to think about a couple of the requirements for your RNA library kit. And this is often determined by the amount of input that you have, the quality of the RNA, and the number of samples, your specific aims, and also your sequencing budget. Do you need to use 2 million reads, or do you need to use 50 million reads? And can you finance that when you're looking at the poor little budgets that we do get for a lot of our research? As an example of the data analysis of looking at the gene expression component here, oops, back here for a sample here. I'm going to show you a 3' sequencing project here. This 3' sequencing project was done with a lot of different samples, so I can kind of show you a quick quality control of what we're looking at here. These are about 48 different samples. We sequenced just the 3' end of these. So we didn't need to use anything more than a small next-seq run. And then when we're looking at the gene expression data here, your gene expression data is the same as if you did a stranded kit, but we just have a lot more samples to kind of parse through. And the nice thing about the 3' expression stuff here and using this kind of tool is that we can go through here and we can start to see the clustering of the samples based on the gene expression data here. So if I was to tell you a little bit about what these samples are, these are actually T cells that have been modified. Some of the T cells are the wild type strains, and then some of them have had CRISPR screens done with them where certain things were knocked out. And then the cells were treated with and without ways to kind of activate them. And with the no treatment versus the treatment here, you can see that we are getting some nice gene expression profiles that can tell us the difference between the purple, which are not treated, versus the green ones here. I didn't add more metadata to this, but I can tell you that there is more than one class of the green samples here. And you can see the clustering up here into kind of small, distinct clusters. And that actually kind of correlates with the CRISPR treatments that we did. And so, you know, this is kind of the beginning part of where we could whittle this down a little bit more to identify whether or not the cells were treated. And then we can go back and do a different comparison between the different subgroups to identify individual genes that we can use for the different treatments that we did on it. From here, you know, we would go and identify qPCR panels to come back into I look at these. So with that, I kind of gave you an overview of microRNA profiling. It's really critical for any sort of microRNA project that you control the samples. Any changes to samples coming into your isolation and library kits will have an impact on your data analysis. And especially if you're starting with anything that's coming from blood, make sure that, you know, you're not having hemolysis. This is a big confounding error that I often see, people reporting these microRNAs are up and down regulated. And when you go back, you say, well, that looks like a hemolytic marker. And I'm willing to bet that if they ran this through, our microRNA NGS analysis portal, and they looked at clustering, they would actually see that the stuff clusters bases on batches of sample, or based on, they could look and actually see hemolysis in the sample if it had, if they knew what they were looking for. For the microRNA data analysis, this is something that we include with our QIAseq microRNA library kits. We want to have a workflow where researchers, as they get started, they can take that FASTQ data and get into the real differentially regulated stuff. And so this is, I showed you one of the pipelines that we have. It's this kind of graphical RNA-seq analysis portal. When you buy your microRNA library kits, you can take the lot number off it. You upload it to the website, you put your lot number in it, and it will give you credits for doing the analysis. What we use the credits for is for doing the primary alignment. Once you've done the alignment, you can go back and you can compare any sample as many times as you want. Your data actually gets stored within that website. So that's why we ask you to log in. So anytime you go through, and if you think about a project, the project I showed you were 6 samples. That was my first 6 samples I had ever done. We used the MiSeq. I was really happy about it, but it's not the end of my experiment. After those 6 samples, we moved up to 24, and then we moved up to doing a couple hundred. Each time I got new samples, I compared those with the old samples. So my first data set was 6. My next data set was 30. After that, my next data set was 360 samples. Each time I kept growing my data set, I was able to go through and compare it with my old data and redo my analysis. One of the nice things about the RNA-seq analysis portal here and showing you this gene expression data is down here in this lower left-hand corner, as we add more data, we can start to look at the pathways that are up and down regulated here. So this allows us to get information from Ingenuity pathway analysis on the samples that we're analyzing here, and it gives us the ability to predict whether pathways are inhibited or activated. We can see upstream regulators, and we can see whether or not this gene expression component looks like a disease or has a particular function based on the pattern that we see. In addition, we can also go through and even filter by some of this IPA data to look at specific genes in a pathway and how that is being represented in our data sets here. So it's actually a really powerful tool that ties together alignments and access to Ingenuity without a subscription. It's basically on a sample-by-sample basis. Lastly, we didn't have a lot of time today, and I didn't get to show you our really cool digital PCR instruments. But once you've gone through and you've prioritized the microRNAs, this is where we think about often switching to that PCR world. And the reason for that is if you've ever done NGS library prep, you know it takes a whole day sometimes. You got to run that sequencing machine. It takes another 12 to 18 hours. I can tell you that the data analysis on our website is extremely fast. Once you have the files uploaded, usually within 4 hours, we can do hundreds of samples and do everything with that differential expression, the Ingenuity report and everything from there. So that part, we've kind of removed any bottlenecks that you may be experiencing. But if you're thinking about a point of care test, there's nothing easier than running a PCR instrument. And our digital PCR platform is as easy as using a qPCR. You don't have to worry about building those droplets anymore. It's basically a plate-based system. You load it into the instrument, it reads it, and you're done within an hour. And you can run a lot of plates really quickly and a lot of samples really quickly. So that I'm going to stop. I can see there's a ton of questions in there, so I'm going to go through and answer as many as I can. If there's any questions I don't get to, I will send an email follow-up with that. And again, thank you for attending our microRNA webinar. MicroRNA is a really exciting biomarker. There's a lot of research going on, and I think it's one of the things that if you haven't started, it's a great time to get into it. If you've already started your research, you know that there's no end to what you can do with microRNA gene expression and looking at molecular mechanisms.
Dr. Rulli, thank you again for your presentation and your time today and for your important research. And before we go, I want to thank our audience as well for joining us today. I'd also like to thank our sponsor, QIAGEN, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for up to 2 years until April 21st, 2024. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.